So my name is William Kindschuh, and I'm an MD PhD candidate in the Korem lab at Columbia University, and I'll be presenting some of our recent work on vaginal metabolomics and preterm birth. So preterm birth is a devastating outcome for both mothers and children, as it contributes to over half of all neonatal deaths, and it can lead to lifelong morbidities. Not only is preterm birth devastating, it's also a common adverse outcome, as it occurs in 10% of all pregnancies. Preterm birth is also a significant source of racial disparities, as it is nearly 50% more frequent in Black women compared to white women. In addition, roughly 2/3 of all preterm births occur spontaneously, with no known cause. So to identify molecular risk factors and mechanisms of spontaneous preterm birth, we undertook a nested case control study in the motherhood and microbiome cohort, which was led by Mikhail Elovitz from the University of Pennsylvania. The motherhood and microbiome cohort was a longitudinal cohort of 2,000 women followed during pregnancy. We used untargeted metabolomics based on liquid chromatography and tandem mass spectrometry to profile the vaginal metabolome of 232 subjects, 80 of whom experienced spontaneous preterm birth. And we paired this data with previously generated 16S microbiome sequencing data. This was a cohort of women who predominantly identify as Black, which allowed us to study spontaneous preterm birth in a population that it disproportionately impacts. So the first question that we sought to answer was, how are vaginal metabolites associated with spontaneous preterm birth? So we identified several strong associations between vaginal metabolites and birth outcome in this cohort. Several of these had higher levels in women who gave birth at term, including choline and tyramine, which was elevated in white women who gave birth at term. We also found several metabolites that were higher in women who gave birth preterm, such as diethanolamine, tartrate, and ethyl glucoside. We were very surprised to discover that these metabolites are likely xenobiotics, meaning that they are unlikely to be produced by the host or by microbes. These xenobiotics do have various sources, but they can all be found in personal care, hygienic, as well as cosmetic products. So to put some of our findings in perspective, it has been previously reported that preterm birth is associated with maternal exposures to exogenous endocrine disrupting chemicals, and our work supports and strengthens this claim by identifying exogenous chemicals in the reproductive tract that are strongly associated with preterm birth. We also observed that some of the metabolite preterm birth associations that we identified may be mechanistically linked. So the graph on the left is from a review showing the metabolism of choline and betaine. And each red point in the graph represents a step that diethanolamine is known to disrupt. Therefore, we hypothesized that in the context of spontaneous preterm birth, high levels of diethanolamine might interfere with choline and its downstream metabolites, such as betaine, which we also found to be higher in women who delivered at term. So having identified several metabolites that are strongly associated with preterm birth, we next asked whether these metabolites were related to vaginal microbes. So to answer this question, we measured correlations between the absolute abundances of vaginal microbes, which are represented here by the green ellipses, and the levels of vaginal metabolites, which are represented by the colored diamonds, with red being the metabolites enriched in preterm birth and blue being those enriched in term birth. So one of our first observations was that metabolites enriched in spontaneous preterm birth appear to have few strong associations with vaginal microbes, which is consistent with our claim that these are likely exogenous metabolites. However, metabolites enriched in term birth, such as tyramine and choline, seem to have several strong associations with vaginal microbes, suggesting that these metabolites may have more of a role in microbiome metabolism. Intriguingly, though, we observed that choline and tyramine, which are two term birth associated metabolites, correlate strongly with several taxa that are typically considered to be bad microbes, including, for example, Gardnerella vaginalis, Atopobium vaginae, and bacterial vaginosis-associated bacterium. So we then asked if we could learn more about the associations among microbes, metabolites, and preterm birth by modeling microbiome metabolism. So we constructed microbiome metabolic models for each subject based on taxonomic abundances, as well as knowledge of microbial metabolism. These models allowed us to predict for each subject the microbial output of many metabolites observed in the vaginal metabolome while taking into account complex interactions between taxa. So we first observed that our models were generally accurate in predicting the levels of biogenic amines, which are a class of metabolites that are believed to be produced by vaginal microbes and which also include tyramine. In each of these plots, the y-axis is the predicted metabolite output based on metabolic models, whose only input is taxonomic abundances, and the x-axis represents independent measurements from metabolomics. So each dot is a sample, and you can see that there is good agreement between the predictions and the observed measurements. So tyramine was both one of our well-predicted metabolites, as well as a metabolite that was associated with birth outcome, as its levels were significantly lower in white women with preterm birth compared to white women who delivered at term. However, there was no difference between these two groups in the microbiome output of tyramine that was predicted by our metabolic models. So this discrepancy motivated our investigation of the accuracy of our models within these groups. So we first observed that the correlation between measured and predicted tyramine was high among the Black women in our cohort, meaning that our models were generally accurate in this subgroup. But this correlation was weaker among white women who gave birth at term, and was even weaker among white women who gave birth preterm. So we next performed various checks to ensure that the reason for these differences in the accuracy of tyramine modeling between groups was not a result of technical artifacts such as model coverage. And based on our analysis, we believe that our models are failing to accurately predict tyramine levels in white women who gave birth preterm, possibly due to a non-microbial interaction or strain level variation that's not captured by our models. For example, various reports indicate that different strains of Gardnerella carry different preterm birth risk, and our models do suggest that they may carry different tyramine production capabilities as well. So we next investigated whether we could use clinical microbiome and metabolomic data to predict spontaneous preterm birth. And this is an important clinical question because current methods for the early prediction of spontaneous preterm birth are simply not that accurate. As you can see here, using just clinical data for the prediction of spontaneous preterm birth yields areas under the ROC curve of between 0.54 and 0.67. And for context, an ROC curve summarizes the performance of a predictor. A diagonal line would be a useless predictor, and we would quantify this as having an area under the curve of 0.5. And the more a line reaches to the top left of the plot, the more accurate a predictor is, with a maximum area under the curve of 1. So cervical length and the measurement of fibronectin, which are two methods that are sometimes used for spontaneous preterm birth prediction, also do not provide high accuracy. So we attempted to train predictive models using gradient-boosted decision trees and robust nested cross-validation. Consistent with other published work, we find that unfortunately models based on clinical and microbiome data perform rather poorly in predicting whether a woman will deliver preterm. However, when we trained and evaluated a model based on metabolomics data, we observe a significant increase in accuracy, in which we achieve an area under the ROC curve of nearly 0.8. So to emphasize, we are taking these measurements in the middle of pregnancy, an average of 3 months prior to delivery, and we were able to make fairly accurate predictions of preterm birth risk. This suggests to us that the measurement of vaginal metabolite levels may pave the way for early diagnostics for preterm birth prediction. So to conclude, we've shown that mid-pregnancy vaginal metabolite levels are predictive of spontaneous preterm birth up to months in advance. We hypothesize that the association between several vaginal metabolites and spontaneous preterm birth may be explained by exposures to personal care products, and we have also shown how microbiome metabolic models can be used to predict metabolite levels and provide insight on the associations among microbes, metabolites, and spontaneous preterm birth. So finally, I would like to thank my co-authors, Federico and Martin, our collaborators at the University of Pennsylvania and at the University of Galway, as well as all of our funding sources. And thank you for your attention. I'm happy to take questions during the Q&A.